In the previous chapter, you learned about microbial growth. Now we're going to take a look at how we can control microbial growth. There's a bunch of terminology in this chapter that you need to know. So here under learning objectives, a big portion is really knowing the terms such as sterilization versus disinfection, antisepsis, degerming, sanitization, what's a biocide, germicide, bacteriostasis, and asepsis. So here, our first terminology slide. Sepsis refers to bacterial contamination. Asepsis means the absence of bacterial contamination. Aseptic surgery techniques would be an example. There you want to prevent microbial contamination of wounds during surgery. Next terminology slide. Sterilization is the removing and destroying of all microbial life. So when something is sterile, that means it is free of microbial life. Commercial sterilizations are common, for example, for canned goods. You want to make sure that even the endospores of something like a Clostridium that could be in the can is removed, because they would grow under anaerobic conditions, and that could be a huge problem, of course. Disinfection is destroying harmful microorganisms, and antisepsis is destroying harmful microorganisms from living tissue. It's important to point out that disinfection does not refer to a specific type of surface. So disinfection could include a wound disinfection. It could also involve disinfecting a door handle or something, that an inanimate surface that where you're suspecting there might be some harmful microorganisms. Antisepsis specifically refers to removing harmful microorganisms from living tissue. Degerming is the mechanical removal of microbes from a limited area. So you want to remove the microbes from a surface that you want to work on. Sanitization means that you are lowering microbial counts on eating utensils to safe level. So if you're sanitizing your silverware, for example, that means you are putting it at a really high hot cycle in your dishwasher, and that will sanitize the utensils. Or baby bottles, for example, that you want to sanitize, you would put them on extra hot in your dishwasher to sanitize them. It doesn't make them sterile, but it definitely lowers the microbial counts. A biocide or germicide is an agent that will kill microbes. Anytime you have the term "cide" at the end, that means to killing. Refers to killing microbes. Bacteriostasis. Stasis means that you are just lowering the growth potential. So stasis means that you're inhibiting the microbes, but you're not killing them. A cide, germicide, will kill the microbes. Stasis means you're inhibiting the growth. Here we have an example for a substance that kills microbes at a certain rate. And just like the growth rate was exponential, the death rate is also exponential, just in the other direction. So here we have a substance that kills 90% of the population per minute. We're starting out at time 0 deaths per minute. Survivors, everyone is a survivor at time 0. And after 1 minute, we killed 90% of the bacteria, and 10% are survivors. And then we're going to kill, out of these 100,000 survivors, we're going to kill 90,000, so 90% again after 2 minutes. And we have 10,000 survivors. And then we have 1,000 survivors, 100, 10, and 1. So you have an exponential death rate right here. And if you were to plot this, it would look something like this. So again, it's a logarithmic death rate right here, so an exponential death rate going down exponentially. And if you plot that linear on a linear scale, then of course you get the typical curve right here, opposite to what the exponential growth would be. And if you are plotting this on a log plot, then you will have a linear death rate going down by a factor of 10 every time. So you have an exponential death rate. How effective your substance is in killing microbes depends on the number of microbes that you start out with. If something is heavily contaminated, it will take longer. It depends also on your environment that you're trying to de-germ. So it could be organic matter that cannot be exposed to high temperatures. Could also be in general the temperature of the environment, whether or not there are biofilms present. All of those things can play a role. Then of course the time of exposure and the microbial characteristics. Any kind of microbe that can form a spore is going to be harder to get rid of than a non-spore former. So, but here are some examples given. So here you can see that it will take longer to get rid of a higher bacterial load. So even if you're using the same treatment or the same chemical to get rid of the bacteria, if you're starting out with more cells that you want to get rid of, it's going to take longer. So here we're starting out with 10 to the 6 cells, and it's going to take us 6 minutes to get rid of all of them. If you're starting out with 10 to the 3, so 1,000 cells, then even this with the same death rate, so the slope of the line here is the same for both, but it's going to take you only 3 minutes to get rid of that lower population count. Let's take a look at how microbial control agents work. There are those that alter membrane permeability and cause bacterial cells to leak out and die that way. There are some that cause damage to proteins, enzymes in particular. Then there are some that cause damage to the nucleic acid, the DNA, or the RNA. One of the best ways to get rid of microbes is heat, because heat denatures enzymes, in general all proteins, and if a cell loses the ability to make to have its metabolism via the enzymes, it cannot survive. There are two terms that you need to know: the thermal death point versus the thermal death time. So the thermal death point is the lowest temperature at which all cells in a liquid culture are killed in 10 minutes. That is something we would have figured out in a lab for different microorganisms if we would have gotten a chance to do so. So you're going to figure out what's the lowest temperature at which all cells in a culture are killed in 10 minutes. So let's say we are putting a culture for 10 minutes at 80 degrees Celsius. Is that going to kill them, or does it take 90 degrees Celsius? So we would have done exactly that experiment in the lab, putting cultures of certain bacteria for 10 minutes at different water temperatures, and then see if we have any survivors, and figure out the thermal death point, which is different depending on whether the bacterium is a spore former or it's more heat susceptible. Then the thermal death point, of course, is a lower temperature. Now the thermal death time is the minimal time for all bacteria in a liquid culture to be killed at a particular temperature. So in this experiment, we would have exposed our bacterial cultures for various amounts of time. Let's say we would have picked 90 degrees Celsius, and we're going to put a culture for 1 minute, another one for 2 minutes, for 3 minutes, 5 minutes, 10 minutes, and so forth on into that particular temperature water to figure out how long does it take for all of the bacteria to be killed at that particular temperature. So make sure you understand the difference between thermal death point and thermal death time. And the destruction or the death of bacteria follows that logarithmic pattern, just like a growth curve. The death curve also follows that logarithmic pattern. And the decimal reduction time, the DRT, is the minutes that will take to kill 90% of a population at a given temperature. A very effective way to get rid of bacteria is moist heat sterilization. The moist heat denatures proteins, so usually you want to bring water to at least boiling temperature and then have the steam flow that then kills the bacteria by denaturing the proteins. Now, maybe the most effective way to get rid of bacteria is autoclaving, and that is we're having steam under pressure. The general protocol is 121 degrees Celsius at 15 psi for 15 minutes, so pounds per square inch, the psi. So that means we are in an autoclave, you have high pressure, and water won't boil until it's actually superheated. It's past the normal boiling temperature, but if you're applying pressure, then water boils at a higher temperature, in this case 121 degrees Celsius. If you are keeping a culture or some contaminated stuff at 15 minutes at 121 degrees Celsius in autoclave, you can be sure that the microorganisms are dead. It will all kill all microorganisms, including endospores, and therefore that's a very popular way of sterilization. So here is how an autoclave works. You have I'm not sure if you saw those when we were still in the lab in the back, the autoclaving all our materials. And once we're done, all the contaminated stuff gets autoclaved to get rid of the bacteria. So we have one of these huge autoclaves, and you put your stuff into the chamber, you close the door, make sure everything is airtight in there. Then you're applying pressure, and that superheated steam, the exposure for 15 minutes at 121 degrees Celsius sterilizes everything. One thing to consider is if you have very large containers that have large volumes inside, then you need to increase the sterilization time, because you need to make sure that the 121 degrees Celsius is reached inside, all the way in the center of that volume that you want to sterilize. We have test strips. I'm not sure if you noticed that when you were given materials that were sterilized, they had some tape on it, and the tape has black bars going across it. So it looks like a regular piece of tape. Before autoclaving and after autoclaving, it has like these lines going through the tape that indicate that yes, now this has reached the correct temperature, and it's now sterile. Here are some examples of how you should adjust the sterilization time in the autoclave based on the volume that you're putting into the autoclave. Take a look at this very large volume right here. This is 6,750 mLs. You're going to have to give it some time to make sure that the correct temperature was reached all the way in the center of that large volume. And now here are some examples of sterilization indicators. We have the one that makes these dark lines to the paper. It doesn't look exactly like this, but like I showed you in the earlier slide, it makes these lines going through the paper. Either way, it's a safe thing to use these sterilization indicator strips that show you whether something has reached the correct temperature. Pasteurization is a popular process for food items. It reduces spoilage organisms and pathogens. It's commonly used for milk and orange juice, just to increase the shelf life of those items. And it's basically a short application of heat. So here are some equivalent treatments given. You could do 63 degrees Celsius for 30 minutes. That's pretty long. Most of the time when food items are pasteurized, you want to use ultra high temperatures for a really short time. So here will be high temperature, short time. That will be 72 degrees Celsius for 15 seconds. Not very popular. Neither one of these are commonly done. But here, ultra high pasteurization, that will be 140 degrees Celsius for 4 seconds. And that reduces a lot of microorganisms that are in milk or orange juice or whatever you're pasteurizing. Some of these thermoduric organisms, they could survive this procedure though. Dry heat sterilization is another popular way of getting rid of bacteria or microorganisms in general. You have the back incinerators in the lab, or we had those. They're still there, but we can't go in there and try them out and work with them. You can use flaming or the back incinerators that we have in the lab. Or you can do hot air sterilization, like basically baking something. And if you have sensitive materials,